All right, today we're playing medical detective. We're going to grab our magnifying glasses and dive into a pretty big mystery. How do you figure out what's really going on inside one of the body's most vital and, frankly, most mysterious organs, the liver?
You know, the liver is a total workhorse. It does hundreds of vital jobs, and it does them silently without a single complaint. But when it's in trouble, it starts leaving behind these really subtle clues. So our mission right now is to follow those breadcrumbs, connect the dots, and solve the case. So let's get started. Okay, so any good detective knows you've got to understand the crime scene before you can even think about looking for clues. For us, that crime scene is the liver cell itself. So before we start dusting for prints, let's get a handle on the two basic ways a liver cell can get damaged. So it turns out liver cells can die in two main ways. On one hand, you've got necrosis. Just think of this as a chaotic, messy demolition. An outside attacker, maybe a toxin, makes the cells swell up and just boom, burst. It throws inflammatory shrapnel everywhere. Then, on the other hand, there's apoptosis. This is way cleaner, much more organized. It's like a programmed self-destruct. The cell gets a signal, it shrinks, and it neatly packages itself up for the cleanup crew so there's no collateral damage. So you've got a messy demolition versus a planned takedown. Big difference.
All right, so now we know what kind of damage we're looking for. It's time to open up our detective kit. And in medicine, our crime lab is basically the biochemistry lab. And our main tools? Well, there are a whole series of really revealing blood tests.
Look, our whole investigation really boils down to answering three key questions. First up, what's the pattern of the injury? Are the liver cells themselves getting hit, or is it more of a plumbing problem in the bile ducts? Second, what's the duration? Is this a sudden one-time attack, or is it a long, drawn-out chronic problem? And finally, what's the severity? I mean, how much damage has actually been done to the liver's function? If we can answer these three questions, we're gonna get to the bottom of this.
And here's how we get those answers. It's actually pretty clever. To figure out the pattern of injury, we look at these enzymes, AST and ALT. Just think of them as the alarm bells that start screaming when liver cells are damaged. For the duration, we measure something called plasma albumin. See, the liver makes this protein, but it hangs around for a while. So if that level drops, it's a huge clue that this has been a long-term problem. And for severity, how bad things are right now, we use the prothrombin time, or PT. It measures blood clotting. Since the liver makes clotting factors, if your PT is slow, that's a major red flag that the liver's function is seriously compromised.
Okay, let's put this new toolkit to the test. Time for our first case, jaundice. This is that classic, unmistakable yellowing of the skin and eyes. It's a major telltale sign that something is definitely wrong with the liver or the biliary system.
So, the culprit behind jaundice has a name, bilirubin. It's this yellow substance that's left over when your body breaks down old red blood cells. Now, a healthy liver, it's like a super efficient filtration plant, just clearing that bilirubin right out of the blood. But if the liver's damaged or if something's blocked, that bilirubin starts to build up. And, well, that's what causes that distinct yellow color. It's a massive piece of evidence for us.
Now check this out, because this is where some simple, kind of old-school detective work is super helpful. If someone's urine and their stool are both a normal color, that points to what we call a pre-hepatic cause. Pre meaning before the liver. So the liver itself is actually working just fine, but it's getting totally overwhelmed with too much bilirubin, maybe from something causing red blood cells to break down way too fast.
Okay, but what if the clues are different? Let's say the urine is dark, but the stool color is totally normal. Well, that points to a hepatic cause, meaning the problem is inside the liver itself. The damaged liver cells just can't handle all the bilirubin, so some of it leaks back into the blood, the kidneys filter it out, and that's what makes the urine dark.
And for our last clue combo, dark urine plus pale stools. This is the classic signature of a post-hepatic problem. Post meaning after the liver, so there's a blockage somewhere in the bile ducts. The liver is actually doing its job processing the bilirubin, that's why the urine is dark, but the blockage is stopping it from getting to the intestines, which is why the stool doesn't get its normal color.
So those simple rules give us a really strong theory of the case, right? And this chart here, this is where the lab tests come in to give us the hard evidence. Now don't worry about all the details here, just focus on that obstructive column. What it shows is that with a blockage, most of the bilirubin in the blood is the conjugated type. That's the stuff the liver has already processed. This totally backs up our theory. The liver did its job, but the bilirubin has nowhere to go except back into the blood.
Okay, case closed on the jaundice mystery. Now, let's tackle another big challenge for our medical detectives. How do we identify the specific viral intruders that attack the liver, things like the different types of hepatitis?
This graph right here tells the whole story of hepatitis A. You can really think of this one as a hit-and-run virus. See that huge spike? That's our old friend the ALT enzyme, the liver's alarm bell. It just shoots up when the infection hits. But then, as the body fights back and makes antibodies, the virus gets cleared out, and look, the ALT level drops right back down. It's a short, sharp battle, and then it's over.
Hepatitis B, though, is a much trickier culprit. I know this table looks a little intense, but it's really all about recognizing different patterns of viral fingerprints. Let's just compare two situations. With an active acute Hep B infection, you'll see the virus's surface antigen is positive. That's a piece of the virus itself. But now look at someone who's immune because of a vaccine. The antigen is negative, but the surface antibody is positive. That's the body's defense shield, not the actual invader. See? These different patterns tell totally different stories.
And then you have hepatitis C. This one can be a chronic intruder. This is no hit and run. This is a long-term siege. Just look at the graph. That line showing HCV RNA, that's the virus itself, just hanging around for months, sometimes years. And see that jagged line underneath? That's the ALT level. It's not one big spike and done. It's this constant up and down, which is a classic sign that the liver is dealing with constant, low-grade chronic inflammation.
So we've looked at quick, acute attacks, and we've seen these long, chronic sieges. And that brings us to the final part of our investigation. What happens when all these injuries just pile up over time? What is the final verdict for a liver that's been under constant attack? And that final verdict, all too often, is cirrhosis. Now, cirrhosis isn't really one single disease. It's more like the tragic end of the road for a lot of different chronic liver diseases. Think about it. After years and years of damage and trying to repair itself, the liver's normal, healthy structure gets replaced by permanent scar tissue. The scarring, it just chokes everything off and stops the liver from being able to do its job.
And this is the really crucial takeaway here. So many of the different culprits we've been investigating, they can all lead down this exact same tragic road. The chronic inflammation from Hep B and C, the damage from alcoholic hepatitis, even the stress from fatty liver disease. All these different attacks, all these different pathways can end up at the exact same destination, the irreversible scarring of cirrhosis. So our little detective story has shown us that, yeah, the liver is incredibly tough, but it's not invincible. A lot of the time, it suffers in silence, and the distress signals it sends out can be really, really subtle. The key, as we've seen today, is knowing what clues to even look for, and then listening carefully when they show up. Which leaves us with this question. Your liver works in silence. Are you listening to its clues?